Continue with chapter 2 of microbiology on what tools we have in our toolbox to study microbes in the lab. Our first order of business from this chapter is going to be, how do we culture them? How do we grow them, get enough sample to work with? And then the second half of chapter 2 will cover the microscope, hence my comic over here on our early microscope, trying to make things bigger, even though it's a woolly mammoth. So a little humor for your day. And with that, we're going to get going. So our first section is on how to culture microorganisms. And while this may seem easy, considering microbes are everywhere, we learned last time that they're ubiquitous, found everywhere. Trying to get the one you want to grow is the challenge. There's a couple others that we need to think about. The first is that they have complex associations with their environment. And since they are found everywhere, in the polar ice caps, in the soil in your garden, on your skin, we have a lot of things to think about to get them to grow. Do they have other microbes that they associate with? What pH, what temperature do they like? And trying to mimic that in the lab can be very difficult. They also have to be grown under artificial conditions. So again, nothing in life biology wants to grow under artificial conditions, and microbes are no different. They have their home, their habitat that they like, and trying to put them in an incubator on a slab of agar is rather jarring. And so it's difficult to get them to grow as they normally would. Another challenge is that they're invisible to the naked eye. So at least when there aren't very many of them, it can be difficult to see whether you have a sample, what it looks like, how many of them there are. And also, as I said, microbes are everywhere, which means it's very easy to get the wrong microbe or the one you don't want into your sample. Contamination is a major challenge in all of science. There is a story from a group of scientists some years ago that painstakingly got a pure culture, and they were trying to sequence this microbe. And then they got to the end and went, wow, that looks a lot like yeast. Wow, that genome, that's super close. And then they realized that they had contaminated their sample by eating their sandwich near their microbes. So instead of sequencing this new revolutionary microbe of a new bacterial species, they'd sequenced bread yeast. So that's just one little story, but pretty much everywhere there are microbes to contaminate and throw you off the trail. And this is a major problem, more than just a sandwich, but for your patients. When you think of making sure that you get the right pathogen with the right antibiotic and the right treatment, it's important that you have the right microbe to start with. Your book gives kind of a roadmap of the steps that you're going to go through to culture and study the microorganism. And very conveniently, it all starts with I. So there's 5 I's we're going to go through. The first is inoculation. So it's going to be taking your sample and beginning the process of culturing it. Then we have incubation. This is going to be kind of your waiting period. So this is going to be when you take your sample, put it in the proper growth conditions, and wait for it to grow and multiply. Then we'll move on to isolation. So this would be when you're picking just the colony of the culture you want and kind of separating the different bacteria. And this one is especially important when we think about patient samples, because most patients, if you take a skin sample or a biopsy or a urine sample, they're going to have multiple microbes in that sample, some that are their normal flora, some that are pathogenic. And so isolating all of them out, separating them, and figuring out which one is the problem is a critical step. Once we've isolated them, then we can move on to inspection. So this can be macroscopically looking like the picture here to the right and seeing, okay, they're round colonies that are kind of orangish. What kinds of bacteria would make orange bacteria that we could look at? So macroscopically, and also giving you a preview of the second half of the chapter, we can look at them microscopically to see what shape the bacteria is. Is it gram negative, gram positive? What sort of aspects can we learn from staining and looking at it zoomed in? And then finally, we'll put all of that together and get an identification, figure out, oh, it's Staphylococcus aureus, it's E. coli, so that then we can move forward either with our research or with treating our patient. All right, so our first step, inoculation. There's a couple of key terms we need to know for this process. The first is to culture. Okay, so you'll hear me say throughout the class, we're going to culture this bacteria for this many hours. We're going to culture it on this type of agar. And basically what I mean by that is we're just growing the microorganism. It's alive, as we've talked about before, so it's going to be growing bigger, getting more cytoplasm, a larger space, and also multiplying the number of microbes. So we're going to be going up exponentially from 2 to 4 to 8 to 16 and so on of the microorganism. The medium is the nutrient that they're grown on. So this would be like if I'm growing it on an agar plate, that would be a solid medium, or we could put them in broth, whatever nutrient they need in our little artificial environment to help it grow. And each bacteria has different specifications for what medium they prefer. Some are more picky than others, but they have very specific things of they need these amino acids, or based on where we find them. Are they in the blood? Oh, well, maybe we need to add blood to their medium, or maybe they're in the soil and we need to think about what other sorts of gases and things like that are available. Inoculation is going to be us implanting or moving the microorganism into the culture media. So this would be if we're taking a patient sample, then we would take from that swab and put it into our broth or on the agar to grow it up. To do this, we will use a sterile inoculation loop. So as it sounds, it's like a little tiny loop. The handle's about the size of a pencil, and on the end of it, there's a tiny loop, about 1/2 inch in diameter. And we can use that to get a little bit of the culture, even if you can't see it. Okay, you can dip that into it and move it to whatever medium you're going to use. Again, it should be sterile. So going back to our last chapter, it needs to be devoid of all microbes, whether they're vegetative and active and growing, or endospores, even if they're going to be dormant or not active. Either way, they have to be completely removed. And one way we do that is with heat. To give you a little preview to a later chapter, we will often flame the loop. So put it into a Bunsen burner, burn anything off, and then let it cool. And then we can have a sterile loop to move the microorganisms from the starting sample to the medium we're going to culture them on. Okay, so once we have inoculated it, we're going to then incubate it. And incubation is going to be isolating your culture in a temperature controlled environment that will encourage its growth. So basically, this means we're going to take our sample that we've prepared and place it in an incubator or in the proper growth environment. So if I want a microbe that's typically found in the human body, I'm going to put it in a 37°C incubator that mimics our body temperature. On the flip side, if I'm looking for a microbe in the polar ice caps, maybe I'm not going to put it in a warm incubator. Maybe I'm going to put it in a fridge or something like that. Whatever environment would encourage its growth. Every microbe has their preferred growth temperature, and so we have to look at it or determine it experimentally to figure out what's the sweet spot for getting our particular microbe to grow and keeping contamination at bay. Growth of the media is shown in a couple of ways. Again, we can't really see the individual microbes, but we can see evidence, indirect evidence, that it is growing. So if we're growing bacteria in a liquid media, this would be a broth. Again, as the name suggests, it looks a lot like, chicken broth or something like that. Can't say I've tasted it, but it does look a lot like what you would expect, from cooking. It's a kind of tannish, clear media when you start. When you inoculate it, let it incubate for some time, you'll start to see cloudiness. So it will become more turbid. You won't be able to see through it as clearly. Sometimes you'll see that there are clumps in the bottom or sediment where a lot of the bacteria have grown up and they've sunk to the bottom. Sometimes you'll see scum or it will float more. Okay, so you'll see evidence there. Sometimes that's also if you have like a mold or something like that, it'll grow on top. And occasionally you'll see a color change as well. All of these would be evidence that while I can't see the individual microbes, I do see that they're growing and multiplying. If I chose to inoculate a solid media, like an agar shown here on the right, where we'd have like a petri dish with some like gelatin in the bottom, we will see individual colonies. So these individual little circular discrete spots are each individual colonies. So when we added the bacteria to the plate, this is evidence that there was one microbe right there, and it multiplied into 2, into 4, into 8, and so on until it has a whole bunch of clones of itself. And each of these is evidence that we had growth from our original culture. A little bit more on the media or the food that we're giving our microbes. It provides nutrients in the lab, again, because we're using an artificial environment. And one of the big goals of using media is to get a pure culture. A pure culture is when we have one type of microbe growing on the plate, or we can isolate it and get only the Staphylococcus aureus or only the E. coli and separate the two. You cannot do this in broth because, again, I cannot see the microbes. They may be floating around in it, but I can't pick out as they float by. To do this, I have to use a solid media where I can see the individual colonies. So a pure culture, you can see we have our white bacteria over here. It's all isolated by itself. The yellow, all by itself. The red, all by itself. The alternative to a pure culture is a mixed culture. So this would be like a patient sample where maybe you took a throat swab or something like that, grew it on the plate, and you can see that they're intermixed. We have white colonies and yellow colonies all together on the same plate. Now, that said, you can go from a mixed culture, and I could take that one yellow colony with my loop and move it to a different agar, and then I would have a pure culture. But as it is, they're mixed, they're interspersed between the two. The third option that we try very hard to avoid is a contaminated culture. So this one would have started out as a pure culture with the red colonies. And then, oops, maybe somebody left the lid off, so something floated in from the air. Maybe you didn't sterilize your loop to clean it off, but whatever happened, we ended up with this white, I don't know, it's probably a mold, maybe a bacteria here. You can see it looks visually different. It's a different color, different shape, kind of more jagged. And we can see it's now contaminated. It's no longer a pure culture. There are several different types of media that we can use to get a pure culture and identify what microorganisms are in the sample. We have physical differences in the media, chemical differences, and different functional types. Okay? So our physical ones could be, is it liquid like a broth? Is it semi-solid, kind of more jello-like, or is it solid like at the agar that we can see the individual colonies on? Chemical composition speaks to the recipe. So is it chemically defined? Like it has this many amino acids and this much sulfur and this much carbon in it. Very defined, we can weigh it out on the chemistry scale and put it in and it's the same every time? Or is it complex, something more like a blood agar? Well, there's going to be some variation based on the batch of sheep blood that I used. It's still blood agar every time, but it's not perfectly identical. So composition can be a difference. And then the function. So am I using the general purpose, grow anything and everything just to see what I've got? Or am I trying to separate things using selective or differential media? Or am I trying to count it or enumerate it? What's the goal of my project? So we're going to go in a little bit more on each of these. The physical state of the media. So we can have liquid. This would be the broth. This one is really good if you're trying to get a lot of bacteria to grow really quickly. And you can see that there are differences based on color change, and it will become more turbid or more cloudy the more the bacteria grows. The downside to using liquid culture is I can't pick out different bacteria. So if I have two kinds in the same broth, I can't separate them very well. Semi-solid, so this one is kind of the in-between. It is slightly gelatinous, but it does have some movement to it. This is typically used to see if your bacteria is mobile. So to do this, you would use, it's kind of like a, I don't know, a toothpicky kind of stab, I don't know, tool. And you could take a bacteria, stab it into the semi-solid media, and then see, does the bacteria move from where I put it, or is it only in that stabbed spot? So that's another tool we have. And then we have our solid reversible to liquid agar. So we have our solid agar here, where it would be very hard. We could see individual colonies, but the reason it says it's reversible is we can heat the agar and put it into a different shape. Maybe we're going from a tube to a plate, and it will re-solidify into that new shape. So it is going to be liquid at high temperatures and solid at room temperature. Here's a couple of benefits of using agar. So again, agar showed up in our last chapter. It's made from seaweed. Okay, so it's a carbohydrate, sugar, branched sugar structure. Okay, and it forms in the lab. It looks a lot like a stiffer jello, basically, kind of tannish colored over here. It is solid at room temperature. Okay, so, but it melts at the boiling temperature of water. So scientists can heat it up, sterilize it. Often we make it in an autoclave. Okay, so like we talked about last time, the little like metal box basically that gets at really high temperatures, and we can put it in there, kill all of the endospores, sterilize it, it melts it, and then when we pull it out, we can pour it into whatever shape we want, like a tube or a plate. And when it gets to room temperature, it will be solid, and then we can put bacteria on it and culture them. It's flexible and moldable. So again, we can shape it to whatever our needs are. Do we need lots of little tubes, or do we want a really big plate to get lots of bacteria to grow? Either way, it's flexible. And most microorganisms cannot digest it. And that might sound like, wait a second, that's a benefit. I don't get it. But most microorganisms can't digest the actual agar, but we can put food into the agar. So maybe if your bacteria really likes, I don't know, glucose, okay, you could put glucose and agar together, and the bacteria would eat the glucose in it, but they can't digest the agar, so they have to live on top of the agar, which means ultimately you get discrete individual colonies. Like if we go back here, we get individual colonies where they're living on top of the agar, but the agar itself is not going to be digested.